There are 4 people online. What do we?
Yeah, I said, hey, come on. It's the first 2 letters.
Hey, Isabel, what's the button to make this hide again? Remember that? Hide. Yeah, you know how to do it, right? How do you make this thing go away?
Are you sharing the screen on this device?
Yes.
Oh,
Or can I just like push it to the side and be yours?
do this and then put it
Oh, just
like
move it
all the way
over.
in the corner.
That's what I'll do then. All right, welcome everyone to our guest lecture. Just some announcements quickly, so that to deal with the smoke. So if you aren't comfortable staying in your room tonight, you can bring your girls can bring their beds up in here if you want. And no, guys in the conference room, and then girls can have the respite rooms or the GIS rooms. That's available to you. You don't have to. If you want to, you can. Stay in your room if you want. Well, that's what we can make do for tonight. The clouds are supposed to blow away into starting the afternoon. So by midnight it should be clear. Sunday should be clear, but I guess there's a chance that it might come back on Tuesday. Now this isn't coming from me or even our bosses, but this is coming back from the dean. If it comes back and this is forecasted to come back, they're talking about the possibility of moving classes down to campus. Okay. If that happens, that decision will be made Sunday morning, and you would have by breakfast or shortly after breakfast, and then the plan would be to leave here in the afternoon, arrive in Columbus by dinner time. I do not know any of those details. Okay. It would be for only a day or so, I think. I don't know those details, where classes will be held, where you would stay, where you would eat. Well, that's coming from the dean, that's what the dean wants to do. Okay. So yeah, we would come back. So that's what I was told to tell you. If you have questions, I don't have the answers. Okay. But the general plan would be to leave the island sometime Saturday afternoon, arrive in Columbus before dinner. That's the general plan. All right. So with that out of the way, we'll go ahead and get started with our Thursday night guest lecture. Y'all been here last week, so you know we meet every week. This week's guest lecture is Dr. Robbie Martin from the University of Tennessee. And like all our guest lectures, I'm going to ask Robbie to kind of introduce himself and how he ended up into the position that he is at. So thank you, Robbie.
Thank all of you. I wasn't really expecting to have to introduce myself and kind of give you an idea of how I got here, because it's quite a circuitous route. I got a degree in forestry in 1991 from Louisiana State University, or LSU, and I went to work for a paper company immediately following that, and I worked in the paper industry, managing a paper company's land base that supplied wood to the paper mills. After doing that for about 20 years, I got tired of it. You go through midlife crises, you burn out, you decide you want to take a different direction in life. And so I decided to go back to graduate school. I always loved biology. I was interested in microbiology, so I went back to graduate school when I was 44 and enrolled in graduate school at the University of Tennessee studying aquatic microbial ecology. From that, I started working with Steve Wilhelm, my professor, who does work here at the Stone Lab, who does a lot of work on Lake Erie studying harmful algal blooms. And My career goal was to get an education, a solid education in harmful algal blooms and aquatic microbiology and a little bit of knowledge about lakes and management. Then take my management experience from forestry, land management experience from forestry, and merge those two, and somehow get into the watershed management career. And somehow I got hooked on research, and I'm still working in the research area, and that's sort of how I am here now. If you guys have any questions about that, I'd be happy to answer them. If not, I'll go right into my presentation. Any questions? Okay, so today what I'm going to talk with you about is a research project focusing on harmful algal blooms that we are currently conducting at the University of Tennessee. And we're at the Stone Lab this week as part of that research project. Now I'm a little bit embarrassed to come to the Stone Lab and present background information on harmful algal blooms. We're talking, you guys are the expert, Justin's the expert. Little bit intimidated and embarrassed to do that, but Justin said it was appropriate for me to do so. And so I'm going to give you a brief intro into harmful algal blooms as a way to get started. If I can figure out how to advance the button.
Yeah.
Got it.
Oh, you got it.
Right. So over the past several decades, harmful algal blooms, or HABs, have been an increasing problem in freshwater worldwide. Harmful algal bloom is an extreme and expansive proliferation of phytoplankton that results in cell concentration numbers high enough to disrupt the natural functioning of a lake ecosystem. Now, blooms can cause really foul conditions in a lake, as I'm sure many of you have seen already, but really foul conditions in a lake that can severely degrade or impact the recreational potential of that lake, causing pretty substantial loss of income or economic activity around the lake. But more importantly, a lot of blooms in freshwater are caused by cyanobacteria, many of which produce a variety and sometimes very potent toxins which are released into the water. And because of all of these problems, cyanobacterial harmful algal blooms represent a substantial threat to our freshwater resources and to a lesser degree to human health. Now, given the annual blooms and experience in Lake Erie, Ohioans are very familiar with this phenomenon. But I'd like to point out that this is a global problem, as the pictures from around the world here demonstrate. Excuse me. And while cyanobacterial harmful algal blooms typically manifest themselves as green water, I also want to point out that that's not always the case. I really love showing this slide because the picture is so dramatic. So this is a cyanobacterial harmful algal bloom in Lake Salubria in upstate New York that occurred in 2020 during April of 2020 during the height of COVID, and it really caused a lot of consternation and concern in the local residents. You can imagine, right? COVID descends on you. Their lake turns red. What's the next plague? Hellfire, brimstone, locusts. They were really upset about that. We did a little bit of work on this bloom, and we determined that the causative agent of this bloom was a strain of Sudanabana that was rich in the red pigment phycoerythrin. Now, for a couple of decades, limnologists have kind of recognized and felt that HABs have been increasing in severity and intensity. This seems to be a trend. But all of these conclusions were always sort of qualified with statements as like, well, this increase in intensity, this increase in frequency could be simply due to increase in surveillance, increase of awareness, right? If you look for something, you find it. If you don't look for something, you might not find it. And that's always been kind of an idea in the back of limnologists' head. But a few years ago, a group conducted a study where they looked at old remote sensing records from 71 large lakes across the globe. And they looked at the period between 1984 and 2012. And what they found out and demonstrated is that, yes, harmful algal blooms are in fact increasing in severity, in frequency and severity in the last several decades. Now, the primary causes of harmful algal blooms are pretty well accepted at this point. So blooms are caused by nutrient enrichment that drives eutrophication. And the nutrient enrichment itself is primarily caused by human activity. And I think the best explanation I ever heard about about the cause of harmful algal blooms came from Mark McCarthy, a scientist who studied that. And he said simply, blooms are caused by too many nutrients flowing into water that's getting warmer. And while that might be a little bit of an oversimplification, it really gets at it really captures the heart of the problem, right? The essence of the problem. And in fact, nutrients are so central to this problem that in well-studied systems like Lake Erie, you can predict with a fair degree of accuracy the amount of bloom biomass in the summer by the amount of phosphorus that's deposited in the lake in the spring. An example of this is the NOAA's early season projection for blooms in Lake Erie this year that is put out by NOAA. And they're projecting if blooms go up to the upper range of their projection, you could have a pretty severe bloom, probably more intense than we've had in the last 4 or 5 years. Now, there's a lot of species of cyanobacteria that cause blooms, but the most notorious and problematic species globally is Microcystis aeruginosa. And in this lecture or in this talk, I'm just going to call it Microcystis. But Microcystis is a freshwater colonial cyanobacterium that again, that's colonial by nature. These are the individual cells, and they make these colonies that's usually pretty visible out in the lake. But they are they're a highly variable species, right? So a bloom of Microcystis is typically comprised of many different strains of Microcystis, and these strains can show very different phenotypes from one another. A factor or a point that I'm going to come back to in a short period of time. Now, Microcystis is a notorious producer of cyanotoxins, especially the toxin microcystin. But not all strains can produce microcystin, consistent with the statements I had just made about how variable the different strains are. All right. So microcystin is a cyanotoxin of great concern globally. And it's one of the first to be discovered and really studied. Back in the 1950s, before this particular compound was isolated and its structure identified, it was known as fast death factor because cell extracts from the cyanobacterium from which it was discovered, when injected into mice, could kill mice really, fairly quickly. Sometimes they would die within hours, and this was quite shocking. But we now know microcystin to be a cyclic peptide made of 7 amino acids, and microcystin is really a class of toxin. There's many different forms of this toxin, and they're typically called congeners. Many different forms of this congener. They vary by the types of amino acids in the ring, that sort of thing. But to date, I think we're over about 300 different forms of microcystin are currently known. What's really striking to me is that after over 60 years of studying this toxin, we still don't understand its physiological function in the cells that produce it. That's still, or it remains uncertain. There is some evidence that microcystin protects the cells from oxidative stress, but in my opinion, there's still room for doubt about that proposed mechanism. Something else that's quite shocking is that the environmental conditions that promote its production are still vague, or at least confusing at times. They're not always really consistent. Now, I think it's obvious that because microcystin is a potent liver toxin that is produced by cyanobacterium that occur in fresh water, being able to predict production of microcystin would be really valuable, right? I mean, it's obvious that would be very valuable. But because of the gaps in knowledge that I just talked about, we really have our ability to predict production of microcystin in a bloom is quite limited. So the earliest efforts towards being able to predict microcystin really kind of relied on these equivalency type equations of phosphorus equals biomass equals toxin. Now, if you go back and remember what I said earlier, that in Lake Erie, you can predict biomass from the amount of phosphorus deposited. So that's sort of the same kind of thing. It's sort of the same kind of relation. But while this relationship, the P equals biomass equals toxin, has been very valuable over the years as a way to try and estimate production of toxin, it's really an unsatisfactory relationship, because we know that total toxin production in a bloom is a factor of a number of things. One is a factor of the amount of toxigenic strains versus non-toxigenic strains in a bloom, and things like the effects of various environmental conditions on the rate of production of toxin in those toxigenic strains. And so the use of this formula, or the use of these equivalencies, left a lot of improvement. Left room for a lot of improvement in our ability to predict toxin. And so 4 or 5 years ago, in an effort to improve our ability to predict the production of toxin in a bloom, a new model was developed. And this model took an agent-based approach in which the physiological production of microcystin is explicitly modeled. And let me say, in which the production of microcystin is in a single cell is explicitly modeled. And this diagram that you see here is a diagram of how the model is designed. Now, this model also took into account the effects of various environmental factors like light and temperature and phosphorus and nitrogen, that sort of thing, to be able to make its prediction. And the model then integrates across millions and millions of cells. So it explicitly models a single cell and the toxin production in that single cell. And then it integrates across millions and millions of cells in an attempt to make some kind of estimation of how bloom dynamics occur, or some kind of conclusions on bloom dynamics more largely. And what's interesting in these models is that higher order properties begin to emerge. These higher order properties, things like competitive advantage of one strain over another, competition between strains, and the drift or change in composition of a bloom based on competition between these various strains. And so to a degree, this model can account for or can address the interaction of different strains of microcystin, and it can address how these interactions may influence the production of microcystin in a bloom. And so the big question that we have, I'm finally kind of getting to the heart of the matter of this presentation. A question that we have is, how does nitrogen affect the production of microcystin in a bloom? Now, I know nitrogen comes out of the blue in my presentation. I haven't really talked about it before. I spoke about phosphorus and how biomass can be predicted from phosphorus loading, but I really didn't say anything about nitrogen. The nitrogen is the other nutrient other primary nutrient that drives eutrophication. And nitrogen can limit or co-limit primary productivity in lake systems. And there's an interesting paradigm that exists. A paradigm that's quite well accepted states that microcystin production increases as nitrogen availability increases in the ecosystem. And this paradigm is almost certainly true at an ecosystem or a lake scale. But if you remember back what I was saying and talking about earlier, total production of microcystin in a bloom is a factor of several things. One being the proportion of toxigenic strains to non-toxigenic strains, and the effect of environmental factors on the rate of production in those toxigenic strains. And so the big question that we had was, does this nitrogen paradigm hold true for all toxigenic strains of Microcystis? And it appears as though it does not. So as shown in this figure, we have evidence to show that some strains of Microcystis, toxigenic Microcystis, like 7806 here on the left, increase, or the microcystin content of cells increases with increased availability of nitrogen. This is perfectly consistent with the nitrogen paradigm. Other strains, like 843 on the right, microcystin content decreases with increasing nitrogen availability, which is exactly the opposite of the paradigm. And so the two different responses to nitrogen availability illustrated here represent a toxin phenotype that has been yet to be described in Microcystis. And so what we wanted to know is, are these phenotypes present in nature? Can you identify these phenotypes? Do these phenotypes do they exist in nature, not just in lab rat strains? And if they do exist in nature, how do these different phenotypes influence the total production of toxin in a bloom? Those questions, those two questions are what is driving this research project. And so the research objectives of this project are listed here. And so we are currently in the process of screening multiple strains of Microcystis in the lab to try and classify whether or not they have either an upper or a downer phenotype. If you remember from the last slide, so an upper phenotype is one that increases toxin production or increases toxin content as nitrogen increases in the environment. And downers do just the opposite, right? After we get through classifying or screening multiple strains of Microcystis, what we would like to do is select 2 to 4 strains, half from each of the 2 phenotypes, and then subject them to extensive experimentation to try and identify and characterize the mechanisms that distinguish these 2 phenotypes. In the last year of the project, what we would like to do is to take the information generated in the lab and in some of our lab work and update the agent-based model to insert into it, this agent-based model, a strain-specific toxin phenotype parameter. At the same time, we would like to screen natural populations of Microcystis to see if we can identify these toxin phenotypes in nature. And part of the work that we're doing here this week at the Stone Lab is the first effort in trying to do that. And then the last thing we're going to try and do is basically validate this model. What we'd like to do is take the updated model and apply it to the various mesocosm populations and data sets that we've been studying to make some kind of estimation of gain of predictive ability in the model when you incorporate this toxin phenotype. I need to get something. Right. So what I want to do now is to kind of shift gears a bit and talk about our one-year objective, talk about the screening and classifying of strains and microcystis that we're currently doing in the lab. Now, our objective is to screen 10 to 15 strains of microcystis to classify whether or not they have the upper or downer phenotypes, whether they exhibit that upper or downer phenotype that I talked about. But most importantly, what I want to point out here is that we're using continuous cultures to screen the strains. Now, using continuous cultures to screen these strains takes a really long time, and it is a lot of work. It is really, really a lot of work. But it has huge advantages over using traditional batch culture methods. So in a batch culture, shown on the figure on the left, growth conditions constantly change, and the nutrient concentrations that are experienced by a culture constantly change. And if you're trying to study the effects of nitrogen concentration on a phenotype, continuously changing nitrogen concentrations can be a problem, obviously. So you can avoid that problem by using continuous cultures. In a continuous culture at steady state, cells experience constant chemistry. And using a system like this, you can get a much more refined insight into how nutrient concentrations are influencing a phenotype. And a chemostat is an apparatus used to grow continuous cultures. So in a chemostat, a continuous supply of fresh media is pumped into the reaction vessel, and an equal volume of spent culture is removed from the reaction vessel at the same rate. And an interesting thing that really develops from this relationship, in a chemostat, you can actually control the growth rate of bacteria by simply altering the rate of flow into the reaction vessel. And so the rate of flow divided by the culture volume, the volume in the reaction vessel, equals the growth rate of the chemostat, or of the bacteria in the chemostat. And sorry, I should have had my glass on. And this is a picture of our chemostat set up in the labs. And you can see here the reaction vessels close up, reaction vessels here. We have the pumps here, the reservoirs here, the pumps the fresh media into the reaction vessels, and then the waste is removed and deposited in the waste container in the back here. And we're currently running 9 of these chemostats. Oh, and the cultures growing here, this is actually microcystis growing in chemostats and growing in 3 different concentrations of nitrogen. And it's very clear, you can see the difference in the cell abundance between the two. Now, the experimental design that we're using for screening of the strains is a nitrogen step-down experiment. The way we do is we start the cultures in the chemostats at high nitrogen. Let them, this is kind of obviously an idealized schematic, but we start the cells, we start the culture at high nitrogen, let them grow to steady state. When they reach steady state, we take toxin samples, and then we drop the nitrogen. down to the next level, and we do and start the process over until we have tested the effects of high, medium, and low nitrogen on the toxin content of the cell. Looking at that data, we can then make a determination of whether this strain is behaving as an upper or a downer phenotype. And what I want to show you next is some of our data. So when we started these nitrogen step down experiments, we saw something that was kind of interesting. But to explain or to illustrate what's interesting about it, I first need to describe to you and show to you how microcystis behaves in a phosphorus step down experiment. So this is real data. This is data from NIS 88, Microcystis ruginosa 88. And the phosphorus concentration of the inflowing media is listed across the top, and the vertical lines indicate the time at which the media was changed. And as you can see, this is biology, a little bit of noise in here, but you know, these things don't grow like E. coli. What you can see here is generally you see a fairly smooth reduction in cell concentration as phosphorus declines, exactly as you would expect, right? This is what we thought we would see in the nitrogen step-down experiments. This is, in fact, what we did see in the nitrogen step-down experiments. And this is the exact same strain, 88. And you can see that dynamically changing nitrogen in these cultures really, really perturbs them. That makes them very unhappy, if I can use, you know, kind of a colloquialism. We don't understand why. We don't know really what's going on. But it really, really upsets the state the cell concentration. Really perturbs the cell concentrations, and we have seen this in every strain that we've screened so far. And some of them behave even more erratically. This is a strain isolated from Lake Erie in 2019 that we got from the Western Lake Erie culture collection. Now this is really causing us problems. If you remember in my schematic, the x-axis went to about 30 days, right? So we thought we could screen one strain in the chemostat in 30 to 45 days. And you see what it's taking us. It's taking us more like 120 days to screen one strain. So this is really, really causing us a problem. So now I'd like to move on to, well, I wish I could have shown you toxin data from those experiments, but we don't have that back yet. We were talking with RU students earlier, and I think that's the bane of existence sometimes in scientific research. Where's my data? I want my data yesterday, right? What I'd like to do now is you move on to your 2 objective, which is characterizing the mechanisms that distinguish the phenotypes. Right. So we have 4 mutually non-exclusive hypotheses that might possibly explain these opposing toxin phenotypes. There could be genomic differences that explain these different phenotypes. There could be regulatory differences that explain them, metabolic differences, or perhaps syntrophic differences. And we think that we can address or test 3 of these hypothetical mechanisms with one experimental design. So the way we're gonna approach this is take those selected strains, 2 to 4 strains after they have been screened. Select 2 to 4 strains, half of each showing the different phenotypes, and subject them to a nitrogen step-down experiment from high nitrogen to low nitrogen and then back again, and while sampling intensively across the entire time point. We're going to collect samples for RNA-seq, for metabolites, trying to think what else we're going to be collecting. For community profiling, for syntrophic analysis. And we hope from that we are able to identify perhaps the mechanism. Now, for the 4th hypothetical mechanism, that being genomic, we're going to resequence each of the selected strains for a detailed genomic analysis. Now, I'm not being naive about how difficult it's going to be to identify these mechanisms. I suspect that it's going to be extremely difficult, very, very difficult. It's one of those situations where conceptually it's simple, it's easy, it's simple, but it's not easy to accomplish. All right, now for the objectives that we plan to focus on in the last year of the thing. We're going to update the agent-based model, screen natural populations, and apply the updated model to basically validate updated model using the data sets from the mesocosms. Now, I'm not going to spend a lot of time on discussing our methods of updating the model because I'm not a modeler, but we have a modeler with us at UT that's going to work on that. Suffice it to say that he's going to take all of the data from all of the from all phases of this project and use that to develop and incorporate a parameter that can address the strain-specific nitrogen responsive toxin phenotype. Now to the mesocosms, the reason we're here this week. Another key objective that we have is to strain natural populations of Lake Erie for the toxin phenotypes using mesocosm scale experiments. And these are two of the students that are here with me this week. So we're here this week trying to accomplish, trying to take a first stab at accomplishing that. Our treatments are really simple. It's the same as we're using in the chemostats, basically. Our treatments are high, medium, and low nitrogen, with low nitrogen being the ambient lake nitrogen as the control. And we hope that we can identify different microcystis strains that show these opposing phenotypes using deep sequencing and single-cell RNA-seq. And again, we know this is not going to be easy, and we suspect that it's going to take multiple visits and multiple data sets before we can actually get this accomplished. All right, so my last slide, the one we've all been looking for. So what will success of this project look like? What will be the benefits of this project? We really have three objectives, or three goals, rather, I think. And that is to characterize a heretofore unrecognized phenotype of microcystis, including the mechanisms that underlie these phenotypes. And then we would like to identify some kind of genomic regulatory markers that allow us to quantify the prevalence of these phenotypes in nature so that they can be fed into a model that can then be used to predict or improve prediction of microcystis. And then the last, we hope to produce an updated agent-based model that includes this toxin phenotype that will improve our ability to predict microcystin production in a bloom. Now, quickly for acknowledgement and funding. I work in Steve Wilhelm's lab at the University of Tennessee, so I have to thank him first, appreciate his support. Then I've been lucky enough to have really good students. Gracie Wren is just a phenomenal undergraduate student. She's done more, as an undergraduate, she's done more work than any grad student in our lab. She's just absolutely an amazing person who is now going off to Georgia Tech to get her PhD. But Gracie has done all of the chemostat work, tremendous amount of work. And then Isabella, Kelly Joyce, and Jason are here with me at the Stone Lab this week helping out. This project was funded through NOAA, through their EcoHab program. And then we also have funding from the NSF and from the Great Lakes Center that are funding Isabella and Jason. And with that, I will try to answer questions if you have any. Wow, that went longer than I thought. Justin, sorry. Yes.
What have you determined so far? Like, like if more of these bacteria are primarily like the amount of waste toxin increases, nitrogen goes up, or do more work the opposite? Like, The more nitrogen there is, the less toxin.
I so wish I could tell you that, but we don't have the toxin data back yet, so we haven't been able to make any conclusions on the strains that we've screened so far as to whether they behave as what I call upward or downward. So I apologize that I can't answer that question as of yet, but hopefully in another month we'll know. Yes.
I was wondering how many different strains you were using and also where you're getting your strains from.
All right, so that's a good question. We hope to we have I don't know, we have about 20 or 30 strains that we maintain in the lab. Most of them are toxigenic. That's the kind we're interested in. And so we're using mostly strains that we have in the lab. We hope to screen about 10 to 15. We had hoped to maybe do more, but it's been so difficult and it takes so much longer than we were expecting. I don't think that we'll be able to screen more than 15. But something that's really, well, you didn't ask that question, so I won't answer the question you didn't ask. So. Well, Justin, if you notice this. So one of our mechanisms that we suspect, and we're building more and more evidence with this as we go, is it might be a syntrophic interaction with the heterotrophic bacteria that feeding nitrate to these cultures. We see some that produce more ammonia and some that produce ammonia and release them into the media and others that don't. And so now we have this production of ammonium issue from heterotrophic bacteria that could be driving, and I won't go into the regulation. All that. But ammonia could be influencing this. But what we have is that the initial figures I showed you, those were conducted with 7806. You don't need to know the strain, but 7806 and 843, they were non-azenic, right? They had heterotrophic bacteria associated with them at the time. We've since been able to get those azenic in pure culture. And we've run those both again as azenic, and we test them regularly, right? We've run them as azenic cultures. And so we have the two most important comparisons that we could possibly make in this program right now. We have 843 with heterotrophic bacteria and without, and 7806 with and without, right? And so that should be the most informative comparison we make. We just don't have that data yet. Yes.
Sites in North America have a history of cyanobacterial blooms, like previously that we're aware of?
Across North America?
Yeah, is that part of the research with?
It's not, but I can tell you, I've talked to some researchers. Justin, you might be able to help me out on this one, but I've talked to some researchers. One that does a lot of work on sediments. And there's history of cyanobacteria, the cyanobacteria die, toxins get buried in sediments. And so you have this stratigraphy that you can go and look at deposition of sediments and all. And they look at sediments and they see various changes in histories of cyanotoxins coming and going in the lake sediments as you go. And some of them are associated with the Native Americans burning the forests. The forests in the southeast were expansive longleaf pine, just millions of acres of longleaf pine that have essentially now disappeared. I was a forester, right? Have disappeared. And there's a guy in Auburn that looks at lake sediments in these areas that were previously longleaf pine, and they see back in the day when they were burned by Native Americans to manage, to manage the understory, they see that they had cyanotoxins. There are lakes I'm trying to remember if there's huge Galpa. Yeah, there's lakes in the mine around mine cities. They had horrible cyanobacterial blooms. So it's not just modern humans, right? The mines didn't probably have really good sewage control out of that. And so it's human activity, not just modern activity, that causes this. Yes.
Question,
Yeah,
sorry. So I know there have been like some studies on mass extinctions with cyanobacteria in Germany and in other places too. And I'm curious if any of your research has suggested that it couldn't just necessarily be human activity, but also certain plant species, like maybe in the Fabaceae family, that are nitrogen fixers that might have a boom in population that might also be making toxic algal blooms before humans were putting pesticides and insecticides on the land, which would be good to know maybe if that could be exacerbating that those scenarios could climax again.
I don't know, Justin, you know, I didn't put you on the spot.
The first global max extinction was due to cyanobacteria.
Yeah.
When they started, when they figured out how to make oxygen, and they started doing photosynthesis, that wiped out all other bacteria around. The first mass extinction was from cyanobacteria. Whole oceans turned bluish green. Yeah, they've been around forever.
Right. Yes. Yes.
I know you understand like the ammonia part of the production that you're seeing inside of ammonia, but do you have any like hypothesis for how the internal structure for that ammonia production might be in the algae?
I should be able to answer that, but I'm afraid I might not be able to. So I know when you supply high amounts of nitrate or high amounts of urea, cyanobacteria. Let me take a step back. So nitrogen metabolism in cyanobacteria is regulated not by availability of nitrate or urea or things like that. Overwhelmingly, nitrogen metabolism in cyanobacteria is regulated by the presence of ammonia. Ammonia is a reduced form of nitrogen, so it takes less energy to incorporate it into it. It's their preference. It's the preferable nitrogen source. And so when ammonia becomes available in the environment, they shut down the use of nitrate, urea, and those type of things. When ammonia is not in the environment, it's kind of an all or nothing with most cyanobacteria. When ammonia is not available, nitrate receptors and nitrate reductases and things like that are wide open, right? They're like, we don't have our favorite lunch around, and so we're going to open it up and upregulate nitrogen metabolism or the use of other forms of nitrogen metabolism. And when you feed them high amounts of urea, you supply them, when you supply cyanobacteria, these guys who with high amounts of urea or nitrate, they tend to dump out excess ammonia into culture. Now you've got ammonia that could be potentially kind of a negative feedback loop. That ammonia could be influencing things. And then heterotrophic bacteria, I'm not sure exactly where they are participating in this. But a number of people have shown in non-azenic cultures, when you feed supply cyanobacteria with high amounts of urea and other nitrogen sources, that ammonia is released. And we've seen that in our chemostats too. When we run up really high amounts of nitrate, they dump out a lot of ammonia, even azenic cultures. You see that, Justin?
I haven't looked at that.
Okay. Any other questions that you guys might have?
Let's thank Robbie one more time.
Let me check and see if there's anything online. Is there anything online? No questions from online. All right, I want to thank everyone for attendance online. Next week, what's the speaker's name? Yeah, next week's speaker is Brian Zimmerman's talking about fish in Ohio. So look for that email, and we'll see you all next week. Thank you all.